All right, welcome to this explainer. We're diving straight into a truly groundbreaking study recently published in the New England Journal of Medicine. And we are tackling one of the absolute highest stakes battles in modern medicine, treating highly refractory blood cancers. Look, when standard chemotherapy fails, and when a patient's own immune system is totally exhausted, medical science has to get highly creative. So today we're unpacking this wild, innovative therapeutic platform that actually engineers healthy cells into an elite, off-the-shelf cellular army, and is designed specifically to hunt down a very particular notoriously difficult type of leukemia. So, let's just jump right into this central paradox. The disease we're talking about is T-cell acute lymphoblastic leukemia, or TALL. Now, for years, scientists have had massive success using engineered immune cells, CAR-T therapies, to cure B-cell leukemias. But T-cell leukemia, that's been a massive roadblock. Why? Because you're literally trying to use engineered T-cells to kill cancerous T-cells. It's kind of like trying to program a heat-seeking missile to destroy other heat-seeking missiles while it's flying right through a swarm of them. How on earth do you cure a cancer when your very treatment is programmed to destroy cells that look exactly like itself?
Section 1. The T-cell dilemma. The friendly fire problem in leukemia. To understand the solution here, we first have to understand why standard CAR-T therapies just absolutely haven't worked for this specific disease.
And this perfectly illustrates the core issue, fratricide. In biology, fratricide is basically the ultimate friendly fire. The target that scientists want to attack on the leukemia cells is a protein called CD7. But here's the catch. Healthy T cells, the exact same cells you are engineering to do the fighting, also naturally express CD7 on their surface. So the moment you program these CAR-T cells to seek and destroy CD7, they look right at their neighboring treatment cells and just attack them. They end up killing each other in the petri dish before they ever even get a chance to fight the cancer inside the patient.
Okay, moving to Section 2, engineering a universal cell, building an off-the-shelf super soldier. To get around this friendly fire, researchers had to pull off what is essentially a genetic medical heist. They needed a cell that could hunt CD7 without actually having CD7 itself.
The absolutely crucial point here comes down to time and health. Traditional autologous CAR-T therapy, that's what you see on the left, harvests cells from the sick patient. But with TALL, the patient's normal T cells are usually already malignant or they're deeply depleted from heavy chemotherapy. Plus, custom manufacturing those cells takes weeks, which is time these patients simply do not have. So on the right, we have the universal off-the-shelf strategy used in this study. Researchers took healthy T cells from a donor bank. Because they're pre-manufactured, they're ready to go instantly. But to make a universal donor cell safe to put into a random patient, well, that requires some extreme genetic engineering.
To build these cells, the researchers didn't use standard CRISPR. They use a highly advanced technique called base editing. Older nuclease-based editing acts like molecular scissors. It cuts right through both strands of DNA. And that can be risky, potentially causing chromosomal translocations or karyotype abnormalities. Base editing, on the other hand, is much more like a genetic pencil. It literally just changes a single letter of the DNA code, a single nucleotide, without snapping the DNA strand in half. It's incredibly precise, which is way safer when you need to make multiple edits all at once.
So, let's look at the specific armor they built using that base editor. They performed a triple knockout, effectively deleting 3 specific genes to solve 3 very specific problems. First up, they knocked out CD7. By erasing that CD7 protein from the cell's surface, the cells become completely invisible to each other. Boom, fratricide problem solved. Second, they knocked out TCR alpha-beta. This removes the cell's natural radar, stopping these donor cells from attacking the patient's healthy tissues, which is a deadly complication known as graft versus host disease. And finally, they knocked out CD52. This lets these engineered cells survive a super powerful chemotherapy drug called alemtuzumab, which is given to the patient to clear out space in their bone marrow. It is just an exceptionally elegant, highly targeted biological design.
Section 3, breakthrough clinical results. Victory in the first battle. So we have this beautifully engineered universal cell, known as CAR7. But as we know, biological theory doesn't always translate into the real world. Let's look at what actually happened when they gave these cells to 9 children and 2 adults with relapsed, highly refractory TALL.
Get this, the study treated exactly 11 patients, and by day 28, 11 out of 11 achieved complete morphologic remission. Every single patient saw their cancer visually cleared from their bone marrow under a microscope. In a high-risk population that had exhausted literally almost all other options, seeing a 100% initial morphologic response rate is genuinely staggering.
Let's break that result down a bit further. While all 11 cleared the visible leukemia, 9 out of the 11 patients, so that's 82%, achieved what is called deep remission. This means the cancer was completely undetectable even using highly sensitive flow cytometry or PCR testing. Achieving this deep, clean state is the ultimate goal because it actually allowed those 9 patients to immediately proceed to a life-saving allogenic stem cell transplant.
And the follow-up data shows the true value of this therapy over time. 7 of those 11 patients remained in ongoing remission anywhere from 3 to 36 months after their transplant. This explicitly proves the clinical concept here. CAR7 isn't necessarily a standalone cure right now, but rather a profoundly effective bridge. It temporarily wipes the slate clean, keeping the aggressive leukemia at bay just long enough to get the patient a brand new, healthy immune system via transplant.
Now, Section 4, Viral Risks and Limitations. The cost of heavy armor. Medical breakthroughs of this magnitude rarely come without a cost, so we have to build a more realistic picture of the therapy by looking at the severe risks and the real-world limitations of this trial. The preparative regimen to get the patient's body ready for these cells is intense. Because the patients receive heavy lymphodepleting chemotherapy, specifically that drug alemtuzumab, their immune systems are just completely suppressed. The hidden cost here is opportunistic infections. The study noted significant viral reactivations, stuff like adenovirus, CMV, Epstein-Barr virus. When you strip the body's defenses to allow the CAR T cells to do their job, you basically leave the door wide open for latent viruses to wake up and wreak havoc. Furthermore, 2 patients suffered from something called antigen escape, which we are going to explore right now. We also have to view these results through an objective scientific lens. Look, this is a phase 1 trial with a very small sample size of just 11 patients. There's no randomized control group to compare it against standard care. Also, because this is a bridge to transplant strategy, the long-term remission effects are kind of mixed together. Is a patient cured because of the engineered CAR7 cells, or is it because of the stem cell transplant that followed? Right now, statistically, it's impossible to separate the two completely.
Section 5. How the cancer adapts. The antigen escape mechanism. This is perhaps the most fascinating and definitely the most challenging limitation observed in this study, the way the leukemia actually fought back. Because cancer is never static. It's an evolving, constantly adapting enemy.
Just notice the sheer biological cunning of the disease here. 2 patients relapsed, but their new leukemia cells didn't look anything like the old ones. The cancer had completely dropped the CD7 protein from its surface. This is what we call antigen escape. The CAR7 cells were still patrolling the body, heavily armed, actively looking for CD7. But the cancer essentially just took off its uniform, becoming completely invisible to the engineered cells. Whether the cancer hid the protein through epigenetic silencing, or if a super rare, pre-existing CD7 negative clone just survived and multiplied, the end result is exactly the same. The therapy was outsmarted.
Which leads us to Section 6, the next frontier, upgrading the cellular arsenal. Science never stops iterating, right? Now that researchers know how the treatment works and exactly how the cancer escapes it, they are already drawing up the blueprints for the next generation of base-edited therapies.
Here is the roadmap for upgrading this cellular arsenal. Step 1. Lower those infection risks. Researchers are looking at whether they can achieve these exact same results without using the heavy alemtuzumab chemotherapy, which would preserve way more of the patient's baseline immune function. Step 2. Counter the antigen escape. If the cancer drops CD7, the CAR T cells absolutely need a backup target. Future cells will likely be multi-targeted, hunting CD7 plus another leukemia antigen at the exact same time. Step 3. Immune stealth. By using base editing to disrupt HLA class proteins, scientists are hoping to make the CAR T cells completely invisible to the patient's immune system. That would allow them to persist longer without requiring such intense, dangerous prep regimens in the first place.
And this quote right here perfectly summarizes exactly what you should take away from this explainer today. Right now, CAR7 is not a magical, standalone cure, but it is an incredibly powerful, remission-inducing bridge. It literally rescues patients from the brink of untreatable disease and buys them the crucial window of time they need to receive a life-saving transplant.
We are basically watching a real-time arms race between human engineering and cancer evolution. We edit our cells to be the perfect hunters, and the cancer mutates to become invisible. As base editing technology gets more compact and even more precise, our cellular armor is only going to get stronger.
But the ultimate question leaves us wondering, as we layer edit upon edit, can we eventually engineer an immune cell so incredibly advanced that it anticipates and outsmarts cancer's every possible escape route? Thank you so much for joining me for this explainer. I really hope it gave you a clear picture of just how incredible this science truly is.